Think of color vision like a camera system, okay, where light enters the eye and it will be captured by the sensors, that is your cones, which is present in the retina, and that will be converted into electrical signals, and then further processing happens step by step until the brain will finally see the colors, right? So if you see this picture here, light enters the eye through the pupil, right, and is focused onto fovea centralis. And fovea centralis is the area of highest visual acuity, and there is maximum cone concentration in fovea centralis. That is the center of a macula lutea. And remember, an important anatomical point what you need to remember here is fovea is present 4 mm lateral to optic disc. So this is the optic disc and this is the fovea here. So it is present 4 mm lateral to optic disc, while the optic disc lies on the nasal side. Here you have a nose and this is the temporal side, right? So this is the nasal side and this is the temporal side. Now while the optic disc lies on the nasal side of the retina where the optic nerve exits. So 4 mm lateral to optic disc we have fovea centralis. Remember, for color vision, we have three different types of cones. Okay, we have red cone, green cone, and blue cone. We have red, green, and blue cones. Clear? Now, our red cone is also called as L cone. Green is also called M cone. Blue is also called S cone. That is depending upon the wavelength. Remember, they absorb the long wavelength light. That's why we call them as L cones. Green will absorb medium wavelength light. Blue will absorb small wavelength light. So we call them as S cones. Remember, the red cones will maximally absorb 570 nanometer wavelength of light. Green will absorb 535 nanometer of wavelength of light. Blue will absorb 445 nanometer wavelength of light. Remember, these values are classically described from the textbook Guyton and Hall textbook of medical physiology. So basics of processing, color processing in the retina. If you see now, we just have three cones, right? We have red, green, and blue. So as of now, remember we have three cones. There are three main wavelengths to which these three cones are responding, and we are able to see infinite colors at this. Now we'll see the structure of each cone. It has similar to rods. It has outer segment, inner segment, nucleus, and synaptic body or synaptic terminal. Structurally, cones are very similar to rods, but functionally they are specialized for color and high acuity vision. Yeah? And the signal flow, if you understand, from the cone, information is relayed onto bipolar cell. From bipolar cell, it will be relayed onto ganglion cell, which will form the optic nerve. Yeah, very simple. From photoreceptors, that is either rods or cones, information is relayed onto bipolar cell. Bipolar will relate onto ganglion cell. But this is not a simple relay which happens. There are some modulation which happens. Okay, there is two cells. Horizontal cell, it modifies the signals between the photoreceptors and the bipolar cell. We have a horizontal cell here which modulates the information. Amacrine cells modifies the signals between the bipolar cell and a ganglion cell. So just to draw this diagram simply now, we have a photoreceptor, that is rods and cones, that will relay information on a bipolar cell. Bipolar cell will relay the information on a ganglion cell. That forms your optic nerve. Now at this junction, we have a horizontal cell that will modify the signals. At the junction between the bipolar cell and a ganglion cell, we have an amacrine cell that will modulate the information. So remember, retina is not just like a camera, it's already doing editing processing which is happening here. So with the help of horizontal cell and amacrine cells. Now that we have understood how the light is captured and passed through the retinal circuit, okay, the next what we need to understand is how the signal is converted into electrical activity. That is the phototransduction which happens in the cones. Okay, then we'll understand how exactly the information is relayed onto the cortex.